So I'd like to thank the organisers for the invitation to talk to you today. I'm probably the one person attending this meeting that knows the least about Lyme disease, and I've really enjoyed the talk so far. I love Fred's talk at 8 o'clock this morning. I thought it was truly inspirational. Stefan's talk yesterday was great. And Michael Cook, I thought the psychosocial aspects of these illnesses was no longer there, but it's clearly a problem. So I'm going to tell you about a grant application that we've just submitted to the Medical Research Council as an expression of interest. So a big thanks to Jack Lambert, and also particularly Janie Quinnigan, who without Janie would never have submitted it on time. So I'll give you a bit of a journey how we came to the application. I'm really excited about the new proposal, which is a collaboration between Oxford, University College Dublin, the Mater Hospital, various research groups around the world, the Lyme Resource Centre in Scotland, and a really interesting company, Soft Cell Biologicals, who I'll tell you a bit about in a minute. By putting this together, I think we've come quite a long way, and it will be a very new initiative in Lyme. And what's interesting about the researchers involved in the project, that none of us have worked in Lyme before, and I think it'll be really fantastic if we can get this funded. So just to give you a summary of what we're proposing, so this won't come as a surprise to most of the people here. Trying to use chronic Lyme as a model for persistent pathogen involvement in chronic disease. I'm interested in other chronic diseases, and I'll talk about those in a second. So the University of Oxford, we're the sort of main applicants. We have UCD, Soft Cell Biologicals as well. And it's a 3-year proposal. This is quite a long-winded process. This is just an expression of interest. If we get through this round, we put in a full application in December, and so it would start until 2022. It's not a small project in terms of the budget, but I think it's pretty a good value for money. So how do we get to this? So back in July, right at the end of July, the MRC put out an experimental medicine call, challenge-led, so human pathophysiology, must be human focused. So most of the project needed to involve humans, need to be ambitious, innovative, and I think we all think our research is that. Experiment driven, could we really sort of focus in on something? But the thing that really I struggled with at the time was that we needed to have an intervention or challenge. And I work on chronic fatigue syndrome, ME/CFS, and really the only intervention that's been around is graded exercise therapy, and there's a lot of bad press on that, and we've also got some data on that as well. So I wasn't quite sure how could we apply for this proposal. And about 3 weeks before, I spoke to Jack Lambert, just a slide about Oxford. So here we are, you can see the department. I work on mitochondria, and actually this is one of the few talks I'm not actually going to mention them, so which is quite surprising for me. So we've been working on ME/CFS for about 3 years, and I'll talk about some of that data later on. So I had a call with Jack back in early July. One of my colleagues in the department, her husband has Lyme disease, and we talked a lot about this because she's very interested in some of the research we do. The call came out end of July. I was just about to go on holiday for August. I've been quite busy, as many of you have during the sort of lockdown phase, but it was too good a call to miss. I got in lots of trouble with my family. But anyway, so we put together a proposal in a really short space of time, and we actually submitted that early September. And it was about 8 weeks from the first Zoom call, so I'm quite impressed with this. And let's hope we can get there. So just to give you a bit of background about ME/CFS, many of you are probably aware of this. I know in chronic Lyme, it's one of the, so until very recently, this wasn't really well characterized symptoms-wise, and some of these things will be the same in Lyme. So people can't carry on their normal activities, profound fatigue. The post-exertional malaise is one of the key symptoms. So if you do anything physical beyond your sort of capabilities, emotional effort, you suffer. You can suffer for weeks or months afterwards. You don't sleep very well. There's cognitive impairments of a brain fog, which I think in chronic Lyme is there as well. This weird thing called orthostatic intolerance. So the symptoms are worse if you stand up, can be less bad if you lie down. And these are the sort of 5 main symptoms that we now look for in this illness, but there's also loads of other things that are going on as well as this. Problems in ME/CFS, and I think in the chronic Lyme field, hearing Jack's talk earlier and the talks yesterday, it's really hard to diagnose. It's really difficult. There's no test. All of the clinical trials, the ones that have been, are pretty subjective, sort of questionnaires, fatigue scores. There's no sort of quantitative measures at all. A huge distrust between the patient groups and the medical profession, and that's been a real problem. And like in chronic Lyme, very little research funding. So we really haven't got going in this space. And one of the things for me, I don't know anybody with ME/CFS, and the human cost is massive, just like in chronic Lyme. So this is a strong family. So I met Nikki about 5 years ago, who's on the left, the mum with the dog. She came to a talk I gave at the local Oxford ME Group for Action, a bit like Fred's thing in Holland, but on a smaller scale. And she said her son Jamie was really interested in science. He'd been diagnosed with chronic fatigue when he was about 17, just about got through his A-levels, started uni, but really couldn't do it. And he's now 8 years on from that. And he's a moderate to severe patient. But you look at him in this photograph and you think, well, he looks fine. And the slides that Fred showed earlier, the patients look normal. And this is a big problem. And the lady on the left is Lara, Jamie's sister. She's really severely affected with this illness. You would have no idea. So that's the thing that it's taken me a while to get used to. Jamie's worked with me for the last 3 years, and I'm going to talk about a lot of his data. So he's been an inspiration, not just as a patient, but also what he can do. And I know what he can't do as well. So that's been really, for me, it's very motivational to see how hard he tries to do the work. So I'm going to talk to you about some of the stuff that we've done in ME/CFS, which we want to apply in this new proposal. This is all unpublished, so please don't tweet it or put it on social media. We're just writing up the papers. It's taken us a long time because the budgets we've had has been really small. And as I go through the data, just think about what we're going to do. So the intervention is a long-term antibiotics treatment. It's not a cure, but we're looking to see whether we can impact on the symptoms and also the biology. So metabolomics is something we work a lot on. I'll talk about that. L-form bacteria, which is the work of soft cell biologicals. These are sort of hidden infections, persistent ones that we heard about yesterday. Raman spectroscopy is a new technique which we've been developing as a potential diagnostic, and we do that in peripheral blood mononuclear cells. And an interesting project we've been running with a private clinic called the Dove Clinic in the UK, who are running a fecal microbiota transplantation program. And I'll mention all of these as we go through. So just to take you through the work we've been doing. So this is the metabolomics. We've looked at 2 cohorts, one from Poland. The group in Poland are actually remarkable. They have no budget at all, and they've done some brilliant work in this field. They diagnose the patients probably better than anyone I know. They don't just do a questionnaire. It's really sort of quite a thorough investigation, and their samples are really pretty good. The other cohort is from the UK Biobank, funded by an ME Association grant and NIH. Bigger cohort, we've got lots of different severities of patients, severe, moderates and milds. We're halfway through working on that, but it's taken us a while. Our total budget for our metabolomics has been about £70,000. And so it takes us a long time, and we have to use the resources we've got. So the group we work with in Oxford is run by James McCulloch, who runs the mass spec unit. And so it's nice to take you through this really. So we get the samples, mainly plasma. We need about 400 microliters of plasma. We process those, and then we run them through the mass spec. The data analysis takes a long, long time, and you'll see why when I talk through the data. And then we're trying to link that to clinical measures, a bit like the talk from, the current chair, Christian, earlier about what they were doing with the different species. Can we link them to phenotype? So metabolomics, what is it? We think it's much closer to the phenotype than some of the other approaches. You can do genetics, you can do GWAS, those type of things, but it takes all those into account: age, disease, the types of drugs people are on. It's really the outputs of all of the influences on a person, which links to the phenotype. So we think it could be really valuable, and that's an approach we've been looking at in many models, not only in ME, but also in some cancer cell work that we do as well. So what are we trying to do with metabolomics? And so what we're trying to do is to see what we pick up in blood in terms of the plasma. How does that tell us about the rest of the body, the cells? So you can see here the horrible biochemical pathways that many of you were taught at college or university. The bit in the middle is the bit I tend to focus on, which is sort of glucose metabolism through glycolysis down through into the mitochondria. So that's my bread and butter. And so what we're looking at are really the outputs from these pathways. They come out of the cell or the tissue into the blood. Can we detect them? And can they tell us about what's going on, on a person, on a whole level? And in addition to this, we've got other factors that influence this too. So not just the tissues, but viruses. Have you got an infection? Fungi? Is there a fungal infection? Bacteria, bacteria in our gut make a huge amount of material. They also produce nasty toxic things that could influence our cells. Did they have an impact as well? Drugs. Fred's drugs that he takes for Lyme, there was that huge box of compounds. They're all going to influence on our sort of physiology. Toxic chemicals, what have we been exposed to? So all these things can impact on what's going on in our bodies. And so these are the bigger organ systems that play a role. So the brain, liver, muscle, heart, and the bowel. But also you've got the microbiome, and we mustn't dismiss that. And I think we're becoming more aware of what influence that can have. And so we're looking at all of these components together. So if you've got ME or even chronic Lyme, you go to the doctor, probably been going a few times because they've not really been able to help you. And there's a few tests, maybe there's 60 tests that they can do. They send the blood off for testing and it comes back nothing wrong. They can't pick up anything that's wrong with you. So what we're trying to do with metabolomics of the plasma is to really look into detail about what's going on in the blood. What can we pick up? So this is just a schematic really of what we're doing. So on the left, you can see the mass spectrometer, and the output looks a bit like that spotty graph in front of it. So at the moment, we can find things that we know in blood. There are things that we can identify almost accurately. We're not quite sure, sort of putative, but there are many unknowns that are real, and they are there. And you'll see as I go through the rest of the talk, these are the key things that we need to find out more about. We get breakdown products and noise, and we need to work out what we're looking at. So what you're trying to do is to work out what are the drivers of an illness in a blood sample. Can we detect them? Because there'll also be things just associated with being ill, and they will be there as well, but they won't be the drivers. There'll be things that will come up as well, and we're going to pick up all those things going forwards. So this is to give you a flavor of what the outputs look like. So just to briefly, don't worry too much about this, but big numbers on here, and you can see why. So we have 4 methods that we use to look at plasma. So we take a 400 microliters of plasma, we split it into 4, we process them slightly differently. So we enrich for different fractions because things like lipids don't turn up in all the fractions. So we have an amino acid fraction, 2 types of lipids, and we have a TCA cycle method to pick up those things around metabolism. We tend to use that more for cells, so if we're looking at cells. So if we look at the amino acids as a good example, we pick over 11,000 signatures on the mass spec, if you remember that little square with the little dots on. 59 are known, 29 we can get quite close, but the bulk of them are unknown, thousands are unknown. And there are some overlap between these groups, so there will be things that are found in all the 4 groups. So we're really picking up about 400 molecules of the 12,000 or so that we think are there. Metabolon, many of you may have heard of, are the sort of world leader in this type of analysis, and most people send their samples to Metabolon. And they'll have a general panel of 768 metabolites, and they've got quite a comprehensive lipid panel, but they're still not picking up all the metabolites in a sample. And you'll get your data back and it will say, these pathways are impacted, but you're only probably looking at a small fraction of what's there. So the group, I can't really see, I've got all the pictures on there. I'll just move this out of the way. Kind of straighten that down. Okay, so on the right-hand slide, you can see the people that have done the work. So James McCulloch, who heads the chemistry, Joe Harvey and Tom Ranger are 2 placement students we have in the lab, and Jamie has done most of the analysis, and Powell from Poland provides the samples. So this is an example of an output. So this is the amino acid method, which is probably the one that's looking the most interesting. So this looks complicated, but it's not really. So what we're trying to see is, with all those thousands of variables, are there variables that can discriminate the 2 groups? So we do a sort of a multivariate analysis, which is shown on the graph on the left. So you've got the red dots, which are the controls. So each one of those dots is a patient, and the green dots are the ME. And you can see just by eye that they really separate apart pretty well. You need to do some stats on this to really make sure that's significant, because sometimes you can get what looks like good separation, and they're not. So the Q2 value here, shown in the middle of 0.7, really suggests to us there's something different in those data sets. And what we do is what's called a VIP ranking, which is a combination of statistical analysis, p-value type approaches, to work out what are our top compounds that are giving this separation. So you can see here, there's lots of numbers, and there's about 5 compounds that come out. And this is telling us that the bulk of the things that we identify that are significantly different between the two groups, we can't identify. So that's interesting on one level and bad on another, that we can't get that much close at the moment to what's going wrong. So if you look over here, interesting from this group, the amino acid method, most of the things that are different are lower. So they're lower in the patients, they tend to be low. There's a couple of ones that go up, but generally they're lower. And these are all pretty significant. I'll come back to this in a minute. Actually, this graph is a good one to show this. So you may have come across this type of analysis before. These are volcano plots. So what you're looking for is significance. So things that are significant in these tests fall out of the volcano, a bit like the lava flowing down the side. So the pink ones are highly significant. So in the amino acids, it's a lot here. The C18 positives, one of the lipids, again, a lot of significant things that are different between the patients and controls. It's nice to see that C18 negative doesn't show much of a difference, because if they're all showing differences, you'd worry. So this is one of our problems. We have too many things that are different. So we're going from a test that you can't diagnose to having over 2,000 metabolites that are significantly different. So this is great on one level, but makes it really difficult to work out what's what, primarily because we don't know what the things are at the moment. So what we're doing at the moment is linking these to clinical variables. So this is an approach we will use in the new study. We'll look at the impact of antibiotics and hope to really try and tease out what are the key variables, what are these unknown, what should we be working on going forwards. I said I mentioned graded exercise is something that's been debated in the UK at the moment. Currently, NICE guidelines is to provide this to patients, and there's been a huge sort of controversy with the patients saying this is a disaster, and they're probably right, it is a complete disaster, and this is being reviewed at the moment, a bit like your tests in Lyme disease, which don't really work. So this paper came out in 2011 by Peter White, showing that graded exercise and CBT could be helpful in patients. Another study came out in 2015, which following this up, again pushing this, and this has been used to treat patients in the UK. The ME Association and other patients really were quite incensed by this. They couldn't believe that this could work. From talking to the patients, where people that have done this had suffered for months or even years afterwards. And what's come out of this is that the data was made public. It took a lot of work to get it out, and it's been reanalyzed, and it was really quite flawed. So we have the ability in this cohort to look at this. So when PAL did this study back in 2013 to 2016, graded exercise was the method of treatment in the UK, in other countries. So patients were getting this. We wanted to see, well, what did it do? So these patients that complete this program, and some did, some didn't, they did it for 6 months. And so they did improve their outputs and their physical abilities, but 50% of patients failed. They either didn't start or they failed. So really, it really compromises certain people. So if you look at this graph, which is slightly different from before, so again, you've got the baseline measures, so the controls for the patients, a robust model, as I said, 0.272 on a Q2 is pretty good. Very high numbers of metabolites that were different. These are our top ranked compounds. So these are the best ones we have. All but one are unknown. But you can see clearly that there's a huge difference. So the controls are the ones in red. So this is based on abundance. This is the abundance of the molecules in the sample. The green are the ME patients here. And it's a blue and red. It's very different. So again, they're on the low side. You can see that they are on the low threshold level. So the question is, if we do graded exercise and they complete it, what happens to these variables? Because one of the questions we've had, or criticisms, these things are different. It's just because the patients are deconditioned. They're not doing much, they're not active. So this is just a deconditioning thing. It's got nothing to do with the disease. And so what do we see? So the first thing to mention is if we do this type of analysis with the cohort after exercise, we see exactly the same. There is no change. We still see a hugely significant difference between them. This is a comparison of the baseline and the ME patients. So the question is, what changes if they complete exercise, we get a really poor Q2 statistic, suggesting the model, there isn't any difference really in the model. And essentially, we only pick up 2 metabolites that change with exercise. So that there is no way that this is a deconditioning effect. And so we're looking to see what these 2 metabolites are, because they change. It could be interesting. And maybe I'll talk about that another time. So to move on really to other things that we're doing, something in the blood. So I mentioned soft cell biological research. They're a small SME in Utah, founded by Brent Hunt. Brent sadly died earlier last year. It was tragic really for his family and also the company, because they just gone into the next phase of expansion. They've got new labs, there were all the new genome sequencing machines have been employed. So he sadly died. Greg Prince, who's been involved with the company for a while, but more as a president. Greg's he's a bit of a megastar. Some of the things he's worked on over the years. He founded a Virion Systems. They've got the only monoclonal antibody still since 1999 that's used to treat kids with respiratory viral infection. So he's now taken the helm, and they're trying to build up. They're doing a lot of COVID screening at the moment, so they're not really doing any work in this. So they essentially took 2,000 people. Brent suffers with fibromyalgia, he's got ME/CFS, he has brain fog. He's not a scientist, but he just was intrigued. Why am I having all these problems? He's had so many shoulder replacements, knee replacements, arthritis. He really couldn't work it out. So he thought there must be something going on. And so what they did was they set up a company to try and culture L-form bacteria, sort of stealth-like bacteria from the blood. And what they've done is they developed a patented method to culture these L-form organisms. So these are bacteria without walls. And you can see 2 examples here. They look really strange. They're sort of large, quite large structures, and they produce these blebs. Actually interesting to hear Natasha's talk yesterday about these biofilms. I don't know, could these blebs be involved in biofilm formation? You can imagine if you've got this in a cell, then these blebs will cause all sorts of problems, but these also could exist in the blood, another type here. And so these have been identified by whole genome sequencing, and they've actually cultured them, and there's over 1,000 of these organisms that they've got in their lab. They also find them in the blood, which we've seen during this meeting. So this cell here, I don't know what type of cell it is, but it's got these L-form organisms inside. This is a blood smear of red cells, and you can see these pathogens emerging from these red cells. So soft cell are going to be a key partner in this project if we get funding. The other thing that they've done, I mentioned, is this L-form culture. So 2,000 samples now from different conditions, multiple sclerosis, ME/CFS, fibromyalgia. And this is a graph showing the average number of L-forms they can culture per person. And essentially, in these chronic illnesses, you're able to culture a lot more pathogens. So in ME/CFS, they're culturing on average over 8 pathogens per patient. If you add in fibromyalgia, the numbers are higher. I don't have any error bars on this. I will get that for the next proposal we get through. 217 healthy samples. So generally, a healthy person will have organisms. So their blood is not sterile. They have got culturable organisms, but these are much higher when you look at chronic disease. And this is over a lot of samples now. So the other thing we're going to do in the new project is some work we've done with Raman spectroscopy. So this is Wei Huang and Yabao is a postdoc in the group. And so what we do with Raman is we're using this approach to try and fingerprint cells. So you excite the cells with a laser or a chemical. So here's thiamine, uridine. They've got quite distinct Raman spectra, even though they're quite similar. So we excite a cell with a laser, and you get a Raman scatter. And it's that Raman scatter that we try and pick up to give us a sort of a fingerprint of a cell. So essentially, if you do this on a cell, you pick up lots of stuff. But it's all quite mixed up. So it's not a really accurate way of telling what's going on in a cell, but it will give you a fingerprint and a signature. So your lipids, your proteins, carbohydrate, things like glycogen have got quite good spectra. It's been used mainly in bacterial systems, and we're using it in eukaryotic systems for the first time. It's label free, so this is really useful. You can do it on fixed material, which is really useful for working with blood samples. It's not invasive, but you can also label cells. So heavy metal, heavy water, and you can look at carbon metabolism. So if you've got a single cell system and grow them in the lab, you can do that. And the idea is we've used it to try and look for physiological states. So we published a small paper back in 2018, just going through the technology. And this is new data that we're just writing up for publication. So what we've done here is we compared 3 groups, healthy controls, the ones in green, ME in red, and multiple sclerosis. Because one of the criticisms I've had when I talk about this work is, well, just comparing from controls and ME doesn't really mean anything. So we picked another control group that has quite a high levels of fatigue, quite different. And what was quite surprising, so we see big differences. So what we've done here is we've taken these graphs, which are really complicated, and we've used a machine learning approach to try and work out what is it about a healthy control that is unique? What is it about an ME patient that's unique? And can we then use those properties to see whether we can identify new samples that we don't know what they are? So you have a training set of 75 of each, percent of each, and then you come in with another 25 and you try and work out which group they're in. And you can see quite clearly here, using this statistical approach, that there is really good separation. So the healthy controls are the green here, they separate from the ME quite nicely, there's some overlap. But what was surprising to us was how different the multiple sclerosis ones were. We didn't expect them to be that different from the other 2. And over here you can see where we are with the sensitivity. So we can pick up ME CFS using this method at 86% accuracy from all the other groups, which is quite striking. We're doing some sort of making sure we're not overfitting this at the moment. It's going to be probably a little bit less than that, but it's a pretty powerful technique. So in the new project, we'll use the antibiotics, and so the question would be, where do these patients go? The Lyme patients, are they different to start with? And if they are, do they move back into the control group when we treat them? And the other thing is, what are we looking at in these PBMCs? There's something in there that's different. Is it bacteria? Is it metabolites? What is different in the samples? And on the new project, what we will do, we'll take these PBMCs and run them through the mass spec to see what actually is in the samples. Restoring a healthy biome. Can this help patients? And this study has been really, really interesting. So in ME/CFS, you have a disease type biome. Can we get that back to normal? So this is some work we've done with a private clinic, the Dove Clinic, run by Julian Kenyon. We helped him write a couple of papers. So the first one was looking just to look at the microbiome. And what we noticed was that in not massive study, but in reasonable number, that the microbiomes, the diversity of the bacteria in the bowels of not just ME/CFS, but cancer, obesity, were quite reduced. They were quite low compared to our control group. Actually, this control group, these are actually patients that are no longer ill. And this is very similar to the control group in the Map My Gut data set. So we think this is real, but it wasn't just the ME/CFS, it was across the board. Julian has run another study with fecal transplant of the microbiome, and the results were really quite striking in this paper. There wasn't any clinical outcome measures, but again, this is something we needed to follow up on. And so this is what we've done. We followed up on this, and this has been hit by COVID, unfortunately. So we have 6 patients that have gone through this process now, but this is the one where we have the most data. And the results are really quite surprising. So let's take you through this. So the patient had a nasty viral encephalomyelitis, autumn 2018, ended up with a really nasty chronic fatigue syndrome that lasted for over a year. Was put on, had seizures, was given Keppra, now had FMT, so that's 10 infusions of the microbiome, and got really well. So came off the drug, had no seizures, and he's doing very well after the treatment. 12 months on, there was no symptoms, and I've just heard that they're currently free of symptoms. So what's going on in this patient? Can we get any clues? So we've changed the microbiome, but this is looking at the blood biome, and the blood biome has changed radically. So if you look on the right-hand side, the pre-treatment, this is done by whole genome sequencing. So we're looking at everything, the bacteria, the fungi in the blood of the patients. So we get a good overview of what's going on. High load of bacterial species, and this is what we see in many chronic conditions. Post FMT, the numbers drop down to 46. If you do a sort of more of a quantitative fatigue score, the DePaul score is very good. Very high fatigue levels initially. Post drops down to 46, and at 5 months, it's going way down. They don't have ME/CFS anymore at 5 months. We've got data coming through on the sequencing, but not yet. So this is just showing you the pre-FMT, the orange ones, and straight after. So what you'll notice straight away is that there's been a massive change in what's in the blood. So here, high levels of Acinetobacter, which is an interesting pathogen. We can talk about that maybe in the chat. That's dropped to nothing. This one's dropped to nothing. And these other ones have come up. So the Pelamonas and this one here, the microbacterium has come up. And talking to soft cell, these grey ones, the high grey bars, are things you normally find in controls. So there's been a huge transformation of this patient's blood biome, which correlates really nicely with symptoms. Which, when I spoke to Jack about the antibiotics, if we change the bowel bacteria with the antibiotics, we could see something quite interesting in the blood, and that's what we plan to do. So what are we going to do on the project? We've got quite a lot of objectives. We want to evaluate chronic Lyme patients and see who shows a clinical response to treatment, and also look at ones that don't and compare them. We want to see, is there a change in the blood pathogen load after treatment, and try and link that to measures of clinical improvement. Can we do that? Are there key pathogens that need to change to see something, or is it just a general change? What about the gut? If we treat with antibiotics, do we see a change in the gut? And we're going to look at that, look at the gut microbiome, and again, link that to clinical measures. Is there things in the gut we can change and have an impact? Because what's striking from the FMT, that the gut change had a massive effect on that body's systemic response. Look at immune profiles. We've heard about this during the meeting. This is going to be crucial. You know, what's the status? How do they compare to controls? And what changes do we see after the antibiotic treatment? Changes in the PBMCs, markers. So this is the metabolomics, looking at metabolomics on the blood cells. And again, do we see changes that link to symptoms and improvement? The gut. There's some interesting stuff in ME/CFS, and it's probably the same in chronic Lyme. There is a leaky gut. It seems to be very leaky. A lot of patients have problems with bowel problems. IBS is very high in ME/CFS. I don't know about chronic Lyme. It's probably similar. Do we see a change in this leaky gut? We can measure it. There are markers that we can use. Do they change with antibiotics? L-form characterization. This work will be done by soft cells, so they'll culture the organisms from individual patients. We'll do the sequencing, and we'll do this on each stage. So we're going to have 3 time points: baseline, after treatment, and then a follow-up. So we'll have different things to look at. Can we culture them? Can we culture L-forms from patients? Because we can use those to evaluate antibiotics on a patient-specific panel later on. And we've got these cultures from a patient, say that the regime doesn't really show benefit, then we can go back to that. Soft cell have done this on urinary tract infections, exactly the same thing. Found out what bugs were in the L-form cultures and used that, and the results are quite remarkable. So it does work. The process, this will work. The spectroscopy could be useful. We see it maybe as more as a diagnostic. It's really easy to do. Mass spec, you could never do that as a diagnostic, but you might find key compounds that you could measure. But at the moment, this is one we're banking on, and we can compare the Lyme data sets to the chronic fatigue and also the multiple sclerosis. And a final thing, I need to mention mitochondria. My group is very interested in the impact of mitochondrial variants and how do you respond to a drug, you know, sort of a HIV drug. A lot of these drugs have impacts on mitochondria. Does the genotype of the patient impact the response to, say, an antibiotic treatment? Timescales, if we're successful, we'd start in 2022, so 40 patients and 40 controls. Long-term treatment, these are the drugs that Jack's picked out from his experience. We're not trying to treat the patients, we're just trying to see whether we can tweak the disease. SolveME, in the US, have got a really nice app that goes on a phone, so we can track symptoms over time. We'll do sort of questionnaires as well with sort of fatigue. The initial study will do some immune cell activation, so glycomics, proteomics in Dublin on fresh samples. Whole genome sequencing on the blood, which will go to Utah. If we can get, the MRC have a policy of not funding companies, so we've got to get around this somehow. Hopefully we can culture the pathogens. We want to try and culture things in the lab. This is something I don't have much experience with, and maybe I know some of the people on this conference could maybe help here to give us some guidance on how we do that, because I'm a little bit not sure what to do. This is about £400,000, this first part, and we could do that initially and then try and get the money for this. Obviously, we'd like all the money in one go. This is the second 2 years. Look at the leaky gut, Raman, Metabolomics, how do these things change? Cytoff, we've got a really good panel now for over 40 immune cells. We can look at those. MAT function, one of the guys we work with is an expert, and did a microbiome. all the investigators, big list. I thought I'd show you the pictures because that gives you a bit more of a flavor for what we've got. What's nice for me, I've put in 2 big grants to the MRC on chronic fatigue, and we struggle to get any clinicians that were really interested. I've got clinicians in Oxford that run the fatigue service, but they're not really interested in research in ME/CFS. So it was fantastic to find Jack and Janie to help with this. Peter Gorman also is a clinical medicine expert. So I'm really excited to be working with a team that are really into the area and into the research. And so I think it's going to really work. Greg on the L forms with soft cell, they're sort of a really important interface. And in Oxford, you know, we've got some good people in Oxford. So we have mass spec, Raman. Steve Taylor is a really good bioinformatician. We use the Zagami, which is worth looking at if you look it up. It's a really good platform to look at visual data. Some of the stuff that Stefan talked about yesterday could be fed into this. It's really useful. Brad is an ex-colleague of mine now in Tasmania, works on stroke. My mitochondria interest could be interesting, and then these people here. I mean, we've got an immune cell component, which is not something we're strong on. Over 8 RAs, nurses and technicians. So I think that's quite good value for this over 3 years. So let's see what happens. So this is the project, 1.6 million, different stages. We find out in November. Stage 2 in December. We've got 6 weeks again to turn it around, but I think we've got the bulk of it now. It's just the detail. Final decision next year, and we try and start towards the end of 2021. Scenarios, they fund all of it. From Jack's talk earlier, I know he's a bit skeptical, but maybe they will. They may not fund the bit for soft cell, so we might need to find 100K. They don't fund any, then we're in real trouble. But I think we've got a really good project here and some great people to work on it, and I'm really keen to get it together. Jordan, how long have I got left? Can you hear me? Hello?
Yes, you are finished officially.
Okay.
For lunch.
Okay.
But we want to take a few questions. I'll just let Christian come on.
Yeah, I've got a few more slides, but I can stop now, or I've got 5 minutes. Because I thought this, you can decide whether you want to hear this or not. So I worked on cancer for 10 years. We have a very similar problem with people not believing what we're doing. There's been a real turnaround in that. We've got drugs coming out of what we're doing that could potentially be new antibiotics. So I could mention that, or we can have questions. So
maybe you could present, because it's very interesting, and if you do it shortly.
I'll do it as fast as I can. I do tend to talk a bit fast. Okay. So we started in 2010 looking at cancer from an energetic point of view. We were looking at the Warburg effect, which many of you know about. The idea that cancer cells don't use mitochondria. They're either mutated or they shuttle things into lactate. So we tried to use this to drive cells to use mitochondria. It failed miserably. We didn't find any effect. But what we did find that mitochondria in cancer cells were really important. But we really didn't even bother to apply for any money because people wouldn't have taken us seriously. Couple of points, the glucose concentration of culture media is really crucial for doing. I remember, so Stefan, we talked about macrophages yesterday, and I know the immune cell, there's a sort of understanding of what cells do, but the glucose can really influence how cells function. So think about media. Oxygen's another one we work on a lot. So we tend to grow all of our cells in a lab at 20% oxygen. It's a long way away from cells in the body. If you're working in a biofilm, it could be even lower. If you're working in a joint, it's even lower. So that's something that we take on board, and it has a big impact on our experiments when we do this. And so trying to culture things from the blood, I'm sure a lot of you have grown things under more hypoxic conditions. We do things wrong in the lab. This doesn't go down very well when I talk to people in my group and in the department. We throw things in glucose that's got the concentration of Red Bull and Coca-Cola. It's not what we should be growing things in, and it influences the results. Oxygen is essential for life, but at 20%, you're going to generate loads of radicals. So it's really, really toxic. So that can influence what we see. In terms of cancer, we're not just looking at one concentration of glucose and oxygen. We've got a whole spectrum depending upon where the tumors are. And we don't take this into account when we work in research. I think we've always done this our way, is a problem, and we come across this a lot. So, you know, I don't want to change anything we're doing, because I want to compare to what's been published, even though if it was rubbish. You know, but if your culture conditions are wrong, it makes your work potentially irrelevant. So this is the key slide for us, for me really. So this is a cancer cell, very glycolytic, growing on different concentrations of glucose. If you look at the mitochondrial respiration on high glucose, which is what everybody grows their cells on, it's really low. The lactate is really high, shown by acidification. But as you switch to more physiological concentrations, then it has a big impact. So, you know, much more mitochondrial function. These cells have mitochondria. They're just turned off when you've got them on too high glucose. So we published a short review with some data, but we haven't really published much on this. I'm going to skip through this because it's not really relevant. So we, at the beginning, 2010, no one really believed any of this. It's all changed in 10 years. There's now many drugs in clinical trials that target mitochondria. We've got a lot of repurposed drugs at work. We were very lucky a few years ago that this company, Navintum, came to Oxford and wanted to spin their company into the university, in which we've done this now. So this is I'm not going to go into too much detail. They've got some really exciting drugs, which are antibiotics targeted at cancer cells. You can see the blue line here. There's a synergistic effect with the drug and paclitaxel. These are tumors from humans, go straight into these mice, and they're really nasty. They grow really fast, but we see a huge impact. But what's relevant to this meeting is that we have over 700 antibiotics targeting mitochondria in our library with this new spin out. It's not spun out yet, but it will be. These L-form properties, I think they've got many properties similar to mitochondria. And there's a growing evident emergence of things in our bodies, in cells. Cancer paper came out in Science not long ago. Bacteria are everywhere in our cells, even in normal people. And so maybe it's just an imbalance of this. And I'll stop there. So sorry to go on a bit longer, but I think this could be really useful in the future.